Thank you very much, and I must say I'm really grateful to be here. Thank you to the organizers for allowing me to be part of this excellent conference. I've learned so much already. I want to acknowledge my collaborators here on this slide. We're part of the University of California Center for Environmental Implications of Nanotechnology. And the collaborators that I have listed here are working together, myself included, to evaluate nanomaterials, first and foremost ones that are in high volume production in an ecological context. So granted, this is a departure from many of the talks that we've heard so far, but I appreciate your forbearance and your interest. So first, just a little more about the UCCEIN. This is the acronym for our center. The idea, it's an investment by the U.S. National Science Foundation and the Environmental Protection Agency, and the idea is that with this considerable investment, that we should be able to learn enough about nanomaterials to help introduce them from an economical standpoint in an environmentally compatible manner. So what we learn should influence how nanomaterials are designed in the future. I should say also there's another center in the United States that's similarly funded, that's at Duke and Carnegie Mellon, but also with collaborators worldwide. So this is the United States investment in trying to understand the environmental implications of nanomaterials. And just to introduce you a little bit more to the center is led by Dr. Andre Nell in nanomedicine at UCLA with co-director Dr. Arto Keller and also Yoram Cohen, Hilary Godwin, and Roger Nisbet. We are a global center. We're headquartered at UCLA and UC Santa Barbara, but we have collaborators throughout the United States, including at other UC campuses, and then all over the world and Europe as well. With a center this size, the expectation and with the types of aspirations that we have and the demand and the mission that we've taken on, the expectation is that we will be multidisciplinary, and we are. And so I won't walk you through this entire slide, but you'll see how the multiple disciplines are working together, hopefully, as I give the rest of this talk. A cornerstone of our center is a rather large nanomaterial library that continues to grow, and I actually am sort of emphasizing this because we start with metal oxides, industrial metal oxides, and I'll be talking a little bit more about those today. These are available in large quantities, of course, but then we also have many of these other nanoparticles that are available in industrial quantities. But what happens within this nanomaterial library and within the center is there's an effort to synthesize different variations according to these various properties and physical characteristics so that we can assess property by property for these various property variants, relationships to mechanisms and physiological and ecological outcomes. So a little bit more about kind of the structure of the center. So what I won't be talking much about, this is how the nanomaterial libraries sort of fit in. It's a large endeavor led by Jeff Zink at UCLA, working closely with Andre Nell's group at UCLA that's performing high-throughput screening to assess in a rapid fashion toxicological outcomes and testing hypotheses that are anchored in biochemical pathways as a function of nanomaterial variants from within this library. And then Yoram Cohen's group in Theme 6, they work to assess the structure-activity relationships that are the output of these two combined efforts. We sit sort of in between the exposure assessment group that is within the ecosystem context. We're informed by this effort, which is it's very similar to the type of work that we were hearing about yesterday, fundamental cell nanomaterial interactions, but inherently we're looking at sort of messier real-world systems, terrestrial, marine, freshwater, knowing and recognizing that we can't possibly assess all situations, all permutations of nanomaterials, and the synthetic nanomaterials with these property variants are rare and expensive. And so modeling is a very important component to help extend the reach of our empirical work, and I'll touch on that a little bit today. So why are we doing all of this? As we all know, the nanotechnology is a growing market worldwide, and I've seen other slides that look like this, and it's sort of interesting to see the emergence of the literature as an index of how fast we're able to keep pace with the growth of the market. But the doubling time for the market is estimated in this article was around a 3-year doubling time. And what's interesting from an ecotoxicological standpoint is, we have a paucity of information over the period of 2007 to 2012 with only 320 entries in Science Citation Index. But the growth rate is actually a little bit higher than for our general information base and nanotechnology overall. And so maybe there's some good news there in that we're starting to catch up in terms of trying to assess environmental implications. So the big picture is the Earth. So in the past 50 years, we have this new sense that the Earth is a system, there are interacting processes, and there's not one perturbation that happens without affecting multiple processes at one time. And maybe we know this very well because of what has happened due to human use of fossil fuels and global implications of that activity during industrialization. But looking specifically at chemicals, decisions that we've made in the past with regards to synthetic chemicals and the toxicity evaluations that we've done in the past have in some cases been short-sighted, and we've been caught off guard and surprised in learning new things like the potential for biomagnification, which wasn't something that was predictable. And so I think, from an ecological standpoint, we really do have to kind of think outside the box because we've been caught off guard before we've been surprised. And the toxicity studies that we've performed in the past weren't necessarily indicative of real problems that occurred in nature. This is also happening, of course, with endocrine-disrupting compounds currently, where in low concentrations, micropollutants can cause insidious effects in populations and communities and ecosystems, for example, downstream of wastewater treatment plants, in this case in Colorado, 2 different plants in Denver, but it's happening sort of worldwide. And this is also something that wasn't predicted. So in the CEIN, we sort of broadly divide ourselves across these big compartments, the terrestrial, freshwater, estuarine, and marine compartments. And the idea, what really drives us, is this concept of ecosystem services. So the services that the planet provides have been monetized. And in a way, it's a way of selling this type of research, this type of focus more broadly to society when people understand broadly that each one of these sort of services that are performed have a value, can be monetized. The break in the line here at nutrient cycling represents the fact that it turns out that nutrient cycling turns out to be the most valuable ecosystem service, one of the highest monetary value, and then food production is another one of high value. So these are the services that ecosystems provide that are of societal not only benefit but necessity, and they have a monetary value. Okay. So nutrient cycling. So what I've overlain here in this case on an agricultural field is just to remind us about some of these major cycles in carbon and nitrogen, and sort of what's driving us as we do this research is thinking broadly about fixation of carbon that would be from the plant population side and mineralization, where, of course, methanogenesis and methanotrophy are also important parts of the carbon cycle, but then incredibly important from the standpoint of thinking broadly about, a primary use in agriculture of fossil fuels is in the production of fertilizer and the application of fertilizer, but to the degree that we can offset this by relying on nitrogen fixation, biological nitrogen fixation, to bring bioavailable nitrogen into leguminous crops, which happens naturally, but also into other types of systems, is really quite valuable. And then, of course, we have these other fates of potential loss for oxidized ammonium, and then the generation of trace gases. I'll be focusing a little bit more on nitrogen fixation as I continue this talk, but when we're talking about that grand ecosystem service of nutrient cycling, this is what we're thinking about, is the environment with these cycles overlaying on it. So the initiation of the CEIN, and we're actually in our 5th year, we're finishing out our first 5-year funding cycle, and at the initiation of the center, we adopted a trilogy of industrial metal oxides to study because these are produced in high volume and they're used in a lot of consumer goods. And these are commercially available. They're studied widely. We've seen a lot of studies this session of this conference already that have looked at these exact materials. And so the way that we approach the ecological research in this center is by focusing our efforts not only on high production materials but also on materials that appear to be particularly active from the standpoint of cellular toxicity, embryonic toxicity, and also bacterial toxicity as assessed through high-throughput screening. So I'm not going to dwell a lot on this work. This is a very important part of the center and actually is of great interest to the Environmental Protection Agency in the United States that has its own program called ToxCast for rapidly assessing multiple different types of chemicals to try and, in a sense, catch up in terms of toxicological data. But you can see the cornerstone, again, is the nanomaterial library with all of these property variants. And you can screen thousands of different nanomaterial variants at once with cell lines and also in high-content screening zebrafish embryo. Again, this is described in a recent review paper and accounts of chemical research by Andre Nell. And it generates sort of a heat map display that tells you which materials and which property variants are causing the greatest degree of hazard to these particular receptors. And then all of this information, because there's so many thousands of data points, have to be analyzed through a risk ranking system. And this is where the computational work of Yoram Cohen comes in. So in a sense, the output of this type of research, which is not what we're doing, but is within a cornerstone of the center, helps to guide where we should focus our efforts from an ecological standpoint. So I want to give just two quick examples of how this actually works from this concept of safe design. So the hypothesis from that work of high-throughput screening was that zinc oxide, in that it dissolves fairly rapidly, would exert its influence mainly on cell lines and on zebrafish through the dissolution and release of toxic levels of zinc ion. And it was discovered that the dissolution process could be hindered by doping zinc oxide nanoparticles with iron, and the biological outcomes were also greatly reduced, as we can see both in the heat map and then also in the direct evidence in the animal model here. So we can, in principle, dope zinc oxide with iron, slow the dissolution, and change biological outcomes. And that was based on a hypothesis that the mode of action would be release of zinc ions, which ultimately are involved in ROS generation and toxicity. Similarly, with titanium dioxide, a different hypothesis, and that is a band gap hypothesis with photoactivation. Iron doping should lower the band gap. That would result in greater biological activity, and we see this in bacteria, also in zebrafish, and we see that primary toxicity to algae occurs because of photoactivity. So the concept of being able to alter nanomaterials to change their properties based on hypothesized mechanisms of effects that are proven and then tested is something that the center is forwarding and making good progress on. We'd like to be able to test some of these synthetic materials that are nanoproperty variants, but first we have to understand what the baseline processes that are occurring in something like a complex terrestrial environment. So the workflow for us has been we start with industrial metal oxides, and the first thing that we want to know is simply, we want to know the effects on plants, plant populations, the effects on microbial communities. There's also a possible another arrow that looks here at microbial populations, but from the standpoint of piecing this together in ecosystem context, microbial communities in soil, which are important in fertility, plant fertility, but also in nutrient cycling in general, and then together the plant-soil combination is an ecosystem in the agricultural context, as I'll show you in a minute, with an agricultural plant that includes plant-microbe symbioses. So the approach that we've taken is to study each one of these populations, these communities, and this ecosystem separately. And then what happens is the output of that information returns us back to now a new set of questions and hypotheses to try and understand why we're seeing the outcomes that we see. So first, some of the research from Dr. Gardea Touriste from University of Texas, El Paso, who's within the UCCEIN, growing soybean, a very important global commodity crop. It feeds much of the world, providing protein, but also feed for livestock. Growing hydroponic soybean in the presence of zinc oxide nanoparticles or ceria oxide nanoparticles, in the case ceria or zinc oxide. The concentrations are on the order of half of milligram per liter to 1 milligram per liter for ceria oxide. And what we see in terms of what happens to the nanomaterials, do they go up into the plant? The zinc is distributed through the plant, but it's actually a zinc ion. It's taken up and transformed. This is under hydroponic conditions. In the case of ceria, ceria is entering the roots. It doesn't appear to be in the plant biomass above the roots, and it's substantially not transformed. But what are the effects on the plants? In the case of hydroponic soybean, the zinc oxide nanoparticles, they reduced root growth, and ceria actually increased root growth. But what was more interesting is the fact that there was evidence of genotoxicity to these soybean plants, in this case grown at 2,000 milligrams per liter of ceria. So then in a separate study, I don't have data here for ceria, but I do have for zinc and titania, and then we'll piece this together in the ecosystem context for soil microbial communities. Studies prior to this one were with carbon nanotubes and also with fullerenes, and with fullerenes showing essentially very little effect of fullerenes on soil microbial communities, no matter what type of assay was performed. But in this case, we used total soil DNA as an indicator of biomass, and we see a dose-response effect. Increasing concentration of metal oxide, either zinc or titania, decreases the total amount of biomass in soil. So this suggests that these metal oxides, when mixed in with soil, are actually bioavailable, which was something we weren't necessarily expecting from the standpoint of previous studies. There were 2 studies with fullerenes that preceded this. And then we also see a change in the soil bacterial community. So there's thousands and thousands of different taxa of bacteria in soil, performing some quite redundant functions, and in some cases narrow functions, for example, in nitrogen fixation, ion cycling, and methane oxidation. These are fairly narrow functions that are assigned to a few taxa, and it was interesting to discover that these are examples, but some of the narrow taxa were the ones that were most susceptible in a follow-up study to the influence of these very common nanomaterials. So I'm only showing titanium dioxide here, but we also have similar results for zinc. So the implication is that so we have plants that are potentially affected. We have soil microbial communities that are affected, both in terms of biomass, microbial community structure, and also in these narrow taxa that are involved in nutrient cycling and ecosystem function. But what happens when we go to a more relevant system at the ecosystem level? And this is where we grew soybeans in a greenhouse experiment. And in this case, yesterday a lot of the discussion was about controls, positive and negative controls. This turns out to be a kind of an involved experiment, and we did not use dissolved ceria as a control. We didn't use dissolved zinc salts or ceria salts as controls. We simply applied 3 different doses of either ceria or zinc to soil, and then grew soybean to maturity. And what you can see here are the beans, so evidence that we grew them to maturity. And then here's a plant that's been sort of deconstructed after harvest. And what you see here is what we hope to see with soybean plants, leguminous plants, are these beautiful root nodules, which is here, nitrogen fixation is taking place symbiotically. The partnership that's formed between the plant and microbe. And then, of course, we have the above-ground biomass, so that's sort of a deconstructed with a beautiful root system and a nodule system shown. What were the effects of growing the plant in the presence of these nanomaterials? So we had essentially no effects on plant growth rate, and this is measured by stem length. It's measured over time during the course of these. These were dwarf plants of growing these plants in the greenhouse. Syria, it turned out that there was a measurable difference in the lowest concentration of growth rate. It's a detail that I don't have time to explain at this moment, but would be happy to discuss with you later. One of the important questions that we had was, besides plant responses, and we have other results that have to do with plant indicators of stress that I won't be showing today, but one of the important questions that we had about plant responses was, where do the nanomaterials go in the plant? Are they taken up? This is a question that people have for the food supply. In the case of zinc, there was a fair amount of translocation of zinc all the way up into the leaves. What we see here is a vascular bundle showing evidence of the brightness of this. Dark field electron microscopy is evidence of zinc accumulation within the vascular bundle, and the EDS confirms that it was zinc. So the zinc is moving up through the roots, from the soil through the roots up into the leaves. And then Jorge Gardea Torrestes, one of his areas of expertise is X-ray absorption spectroscopy, and he used the facilities here in France, in France and also in California to perform this work that was able to tell us what the speciation was of zinc. So just because we can measure by ICP total zinc in the leaves, we don't necessarily know the form, and so the big question is, were there nanoparticles that were moving into the leaves? And the answer here is no. The zinc was substantially transformed as it moved from the roots up into the upper parts of the plant, the above-ground biomass. By the time it was in the leaves, it was mostly in the form of zinc citrate. In the roots, it was mostly in the form of zinc nitrate. So it was actually biotransformed as it moved through the plant. And there were two different beam lines that were used to be able to isolate to a fairly high resolution the localization of zinc up in the stem and the leaves, and then also in the pod itself, in the soybean pod. And another zinc synchrotron result. In terms of Syria, what we found was that by ICP-MS, we found no evidence of Syria moving into the above-ground biomass, but we found Syria in the roots and also in the nodules. And one thing that was an interesting, particularly surprising and interesting finding compared to the control electron micrograph of the infection zone in the nodule. So the nodule that I showed you, those round balls that were on the root, if you take a section of those and then look at under high-resolution microscopy, you should see evidence of infection with these bacteroids, the symbioses that are carrying out nitrogen fixation, which we saw in the control nodules in the absence of nanomaterials, but the nodules with plants that were grown with Syria in the soil were empty, which was sort of surprising. They were empty. The nodules count and the nodule mass was the same as the controls, but there were no symbionts in the nodules. The nodules were there, but they were empty. There was Syria in the nodules, but they weren't really performing at these two higher concentrations nitrogen fixation. So the evidence for the presence of Syria having something to do with the performance was through the simple acetylene reduction technique. That's a conventional technique for measuring the potential nitrogen fixation in nodules. And at the medium and high concentrations of Syria emitted to soil, we saw basically the cessation of nitrogen fixation potential. When Jorge Gardea Torrestes used these X-ray synchrotron methods to investigate if Syria had actually moved into the above-ground biomass. He found something that we weren't able to find with ICP-MS. He found that the soybean bean itself, the actual grain, actually did contain Syria nanoparticles. You can see here the red spots, and then over on this side, the green spots. And the point here is that the best fit is to the actual Ceria nanoparticle. There was also evidence that Ceria was transformed, so there was evidence for both a +4 and +3 oxidation state. So there was mobilization of Ceria into the beans, but then also some evidence of transformation. So the point here is that as when Jorge tells this story so elegantly and goes into the details of X-ray synchrotron methodologies, the point here is that now we have Ceria particles in the bean, which means they could be passed on to the next generation. So that was the implication mainly of this finding. So what we want to do, because, you know, as you can see, this is, when you do a study like this, it's kind of a one-off, and it generates lots of other questions in terms of plant stress indicators, microbial community composition differences, exactly where the plant's getting their nitrogen, if they're still growing but not fixing nitrogen at medium and high concentrations of Ceria. There's a number of different types of data that we've collected that we're in the process of analyzing and will be submitting for publication. But it's obvious that we can't perform this type of microcosm, this type of mesocosm for every single nanomaterial, and certainly not for all of those property variants that are populating our nanomaterial library. So, you know, the question now is, where do you turn to? You turn to mechanistic studies, and so we go back and try to understand why are we seeing the effects of Ceria on nitrogen fixation. And then we employ a modeling approach called dynamic energy budget modeling, which is a mechanistic framework. It's basically a series of differential equations that describes energy generation, energy transduction, energy investment in maintenance in cells and in populations. We use that to extend the reach of our empirical work to basically describe mathematically what we've observed, and then be able to employ those models to make predictions with other types of materials, other types of exposures. So what I want, and then other levels of biological organization, including past the population level and up to the community and ecosystem level. So I just want to show you an example of DEB modeling. It's been used in our center by Professor Roger Nisbet, his group, and his colleagues, now within our marine ecotoxicological work, but I'll show you the work that we've done with bacteria. So a study that we came into the center with was one where we had fed Pseudomonas aeruginosa, an environmental strain of bacteria, cadmium selenide quantum dots. These were citrate-capped quantum dots that partially dissolved, but at the time, during the course of bacterial growth in a normal population growth experiment, well mixed in the laboratory, the nanoparticles didn't fully dissolve. So there were both particular forms of cadmium selenide and then the ions that were shed partially. The control was no nanoparticles, no metals, and then there was also a cadmium acetate control. And you can see that the cadmium was accumulating in the periplasmic space in these bacteria, but not getting inside. They have these cadmium efflux pumps that were getting plugged, and it's common to see cadmium and other heavy metals accumulating in that area of bacteria. With the citrate-capped cadmium selenide quantum dots, there was membrane damage that was observed, there was ROS that was measured, and the quantum dots were actually entering the cells and accumulating. And so, as an aside, we also looked at whether or not trophic transfer could occur, because knowing that the quantum dots had entered the bacterial cells and had bioaccumulated, we wanted to know if their protozoan predator would also take up the quantum dots, and how that would affect the protozoan predator. And what we observed was biomagnification. So the concentration of cadmium inside protozoa was actually higher than it was in the bacteria that harbored, that had bioaccumulated the quantum dots. And this was substantially due to a failure in the digestive process. So the protozoans were gluttonous consumers of bacteria that were laced with quantum dots, and they were unable to fully digest those bacteria. The exact mechanism is unknown at this time, but that was the reason for ultimately the biomagnification of that metal. What we wanted to do was then take these data sets, which were really just population for the bacteria, population growth curves, and be able to model the effects of first cadmium, the positive control, and then cadmium selenide quantum dots, because the conclusion of the study had been that there was a distinguishable nanoparticle effect. That is, the effect on bacteria was not simply due to cadmium, it was due to the nanoparticles themselves that were also present. And we were able to model these population growth curves. The actual mathematical formulation is not shown on this slide, but to suffice it to say that we were able to model these growth curves, both acclimation, exponential growth, and up into stationary phase. At the same time, this isn't just an over-parameterization of a process, it's a hypothesis-driven exercise where one is hypothesizing the mode of action and then formulating the model to express the energy investment in overcoming that process. And in this case, we were able to explain cadmium toxicity to cells, and then we further developed the model taking into account the acclimation required by cells to overcome ROS-induced stress from cadmium selenide quantum dots that were present, and we're able to come up with a best fit model for that. So what we hope to do is to take this approach and extend it into the realm of nitrogen fixation and be able to model at the population level effects, for example, of ceria or other nanoparticles on nitrogen fixation, and then raise that to the next biological level and try to understand what's happening in the plant microbial partnership, which will be complex, but it's something that we'd like to take on. So in summary, what I've tried to explain to you is how we're approaching our ecotoxicological work within the UCCEIN. And again, it starts with having a firm grounding in understanding of cellular responses and embryonic and bacterial responses to nanoparticles, which can be delivered in a rapid fashion because of the high throughput methodologies that are used coming out of Theme 2, Andre Nell's group. We're also forwarding, because it's not always the case that nanoparticles that are found to be innocuous through high throughput screening turn out to be innocuous under environmental conditions. So we're also forwarding by convention high production industrial materials into this system. The modeling is the modeling focus is indicated by the arrows. First, to look at multiple stressors and to look at to model the effects of nanomaterials at cellular and organism and population levels, and then to take the empirical information to refine the model to understand what mechanisms, because the modeling can actually help to inform our hypotheses regarding mechanisms. And then to a goal, an ambition, is to increase the biological organization that is the target of our models of a higher biological organization. The color didn't come out here, but the scenarios that I described to you both for terrestrial and I did not describe for marine or freshwater are then to use these other types of information, both modeling and the lower tiers of information, to drive the mesocosm studies that we do, as I've shown you today. So I'd like to acknowledge the researchers that did this work, and this is a short list of the key people that did the work, but also within a center we all work together. And I'd like to thank my colleagues within the center, and then, of course, the synchrotron facilities as acknowledged in the publications. Thank you.
Thank you very much, and thank you for keeping the time. So it's open for questions.
Renata Berayavak, Dubendorf. Thank you, Patricia, for your talk. I have a question to the cerium oxide study. You mentioned that the nodules were full of cerium oxide nanoparticles, but without symbionts. So my question is, are the bacteria destroyed by the cerium nanoparticles, or they do not enter the nodules? What's happening?
That's an excellent question, very perceptive, and it's exactly our question as well. We don't have the answer yet. It could be that an infection has occurred, but because of the presence of Syria, there's been inhibition of growth of the symbiont. It could be that the infection was unsuccessful, but the nodules still form. And it's difficult to find a lot of information, as we've seen during our investigation so far in the literature, of this phenomenon of empty nodules and what can lead to this phenomenon. But it can either arise from what I understand, those two different types of processes, the inhibition of growth of the symbiont or the absence of successful infection, despite the fact that the nodules still form. So our follow-up studies we're working with Bradyrhizobium japonicum, the inoculum strain that we used in these studies, in pure culture to understand its sensitivity. And then we'll be working with hydroponic soybean under nitrogen-limited conditions to investigate nodule formation and how we can either promote this outcome and further learn why it occurs, or try to inhibit the outcome.
There's another question there.
Thank you for your impressive presentation. I'd like to know about something about Syria also. Well, cerium oxide sometimes is reported to have antioxidant activity, but you find out that it was genotoxic. And can you comment on the mechanism you're supposing that nanoparticles to have?
Well, that's interesting. So why was it genotoxic to hydroponic soybean? I don't even think that in that paper there was a hypothesis about why that occurred. So obviously it was taken up. There was bioavailability. It was exposed. It may have something to do with the redox activity of that particle. It's very redox active, so it could be that there is something having to do with that. It's a very good question. I don't have an answer for you at this time.
There's another question.
Hi. I was wondering, because you said that in some ways this was a one-off, just to sort of see how things are gone. And I have a very ignorant question of why you chose the types of nanoparticles. Was it that they're accessible or easy to work with or cheap or extraordinarily important in the life of the ecosystem? And then the follow-up question is, would you then try to do this with other types of particles?
Yeah, and when I said one-off, I didn't mean to sound flippant, because it was an investment to do that type of study, and yet it's the most realistic type of study that we could do in the agricultural context at that time. And so we do intend to repeat that study, but as we did with this particular study, approach it with a considerable amount of thought and planning. We spent probably 9 months with weekly conference calls with all the collaborators discussing every single detail, from what type of nanoparticle, the concentrations, the justification for the concentrations, what type of inoculum for the nitrogen-fixing bacteria, what type of soil. Every single detail was carefully selected, and we do intend to use this approach again. The reason that we use those metal oxides is because they're in high production, but we also had data from the hydroponics study. And so one thing that becomes important from the soil perspective is this issue of bioavailability. There's been this sort of thought that when nanomaterials enter soil, no matter what type of core or coating chemistry, that they become embedded in the matrix and essentially they become non-bioavailable. But the research that we performed for these particular particles would suggest that that's not the case. There needs to be more research in this area to understand the various types of outcomes.
Any more questions?
Hi, I'm Halska from the Institute of Work and Health in Lausanne, and thank you for your talk. It's very interesting and it's very important, I think, also in this context, talk about the ecological processes. I'm wondering what the center's relationship is with your research in regards to industrial processes. So if you have any relationships with manufacturers, and if you look at the data of disposal rates, for example, and how you take the consideration of industry in the area into the question of then ecological mechanisms.
Well, so there is an effort within the center to get involved, and I think we'll hear some talks this afternoon. I'm greatly looking forward to in the area of life cycle analysis to better understand from a global assessment basis the flows of various nanomaterials. And so there is some of that research that's going on, and inherently that maybe not directly but indirectly tethers us to industry. We become more informed. We've also worked with industry in terms of surveying their workplace practices and their disposal practices. We've worked, there's been a collaboration across social and natural sciences, and we've published two international surveys. Well, the second one was more focused on U.S. The first one was the first international survey of workplace practices and reported disposal practices. We interact with industry in terms of their association with our advisory board, and our aspiration in the next 5 years with the center is to accelerate that interaction so that we can ensure that the research that we're doing is most reliably relevant to what the industry trajectories are looking like. So,
Further questions? This one there.
You mentioned, if I understood, you mentioned that when you were measuring the thing with ICP-MS, you didn't see much, and you needed to have radio label type of measurement.
so with Syria in particular, the resolution of ICP-MS was not low enough for us to be able to resolve Syria in the above-ground biomass. The X-ray synchrotron methods are much more sensitive, albeit not quantitative. So we're able to see Syria using those methods up in the bean portion of the plant, but we cannot quantify by ICP. But we feel that the evidence for the material in the bean is pretty unequivocal according to those methods. We just can't quantify because it was below the limitation of that instrument.
Okay.
Good quantifying the root and the nodule, yeah.
We have also a short comment on your very interesting presentation. What struck me most was the term biomagnification. How substantial is this data? I mean, biomagnification we know from other pollutants. And yes, by myself, I have worked for many years on biomagnification of mercury
Yes.
in different marine and freshwater systems. So have you any idea about the mechanisms which could be behind?
In this case, it was only 2 organisms involved. So it would be nice to continue up the food chain. But I believe, and I would like to talk with you more about this or anyone else that has been studying biomagnification, that in this case we were awakened potentially to the role of prokaryotes in the initiation possibly of this whole food chain process. And in that cartoon that I showed of DDT biomagnification, the suggestion is chlorella is the initiator, which I think is very interesting. We wonder if bacteria are also sometimes initiators. In this case, it was just from bacteria to the protozoan predator, and the evidence was clear, based on analytical chemistry, that the concentration of cadmium was increased in the predator over the prey. There was a process of cessation of digestion, and exactly why digestion was slowed down, we don't know at this point. We would like to follow up on that.
Very interesting, very exciting.
Okay. So thanks again, Patricia, for this interesting talk, and we